Hello, my name is Banggun Shin. I'm in charge of AI at Deargen. I'm happy to introduce our company and our technology about drug discovery to this conference. Deargen is an AI-based drug discovery company, and we become globally famous after we published a work about potential COVID-19 drugs back in January. That is even before the pandemic. Throughout this talk, I will introduce deep learning models for drug discovery. And after this talk, you will understand what kind of method is applied to predict those drugs and how a machine can do it precisely. So this is the outline of the talk. After some introductions, I'll describe drug target interaction task. Our approach requires a bit of background of recent deep learning methods. Therefore, I'll briefly explain some of the core techniques like word embedding, transformers, and other DTI models. And then I'll present the molecule transformer that is used when we predict remdesivir in the COVID-19 project. Before going into the details, I'll touch some basic stuffs in drug discovery, that is how a drug can cure a disease. So here in the figure, we have two persons. One is healthy, the blue, and the other one has a disease with the red. The fundamental difference between these two persons is proteins. A human body consists of many different types of proteins, and if you look at the healthy one, the protein distribution is well balanced. However, if you look at the person with a disease, some of the proteins are off the normal level. So we can make this person back to normal state with a drug. An appropriate drug is able to regulate these protein levels to be normal, which means it could potentially cure a disease. More specifically, if this person takes a drug, then it will attach it to the target protein and it will regulate the number of that proteins. If the drug successfully regulates the protein, this person's disease will be cured. So we understand how a disease can be cured by a drug. Now, the next question is how we can develop such a drug. This diagram is presenting the drug discovery process. A drug can be developed through many subtasks, roughly divided into two parts. The drug candidate generation part on the left and the clinical test part on the right in the figure. The drug candidate generate part occupies about 40% of the total drug discovery cost, as you can verify from the table. And in that part, about 5 to 10 K number of molecules are suggested. However, more than 99% of them are discarded by the second part. So the drug candidate generation part is very costly. Therefore, if we can do this better with the help of deep learning, we can save a lot of time and money. So among many subtasks in the drug candidate generation part, we will take a look at the molecule discovery part by drug repurposing. This can be done using drug target interaction model, in short DTI model, which will be explained in the comparable slide later. So what is drug repurposing? Let's take an example on the table. This table shows some example drugs that is repurposed. For example, estamizole, a drug in the first row, is designed as antihistamine. If this drug can be effective on malaria, then estamizole is repurposed to a new target disease, malaria. Um, the advantage of drug repurposing is safe and cheap because it's already proved to be safe through a clinical test. So we can skip some of the clinical tests when evaluating for a new efficacy. Therefore, we can save a lot of drug development money. Okay, now we go back to the DTI task. Formerly, the DTI task is composed of two inputs, a molecule and a protein. By taking these two inputs, the model is to predict affinity score, which is how much interaction would be between the two inputs. A molecule is represented as either a graph or a sequence, while a protein is represented as an amino acid sequence, because most of the time the 3D structure of a protein is unknown. So our model used sequence formats for both of the inputs and applied transformer-based model to represent a molecule and achieved a new state-of-the-art result in the public DTI task. So we published this result to Machine Learning for Healthcare conference in 2019. As I mentioned in the previous slide, we used sequence formats for both of the inputs. Specifically, a molecule, for example, here, lapatinib, is represented as a combination of atom characters like C and S, and other special characters like equal signs and parentheses. So it's a sequence. Likewise, a protein is a series of many amino acids. Therefore, they are all sequences, just like a natural language text. So the good news is that we can leverage many natural language process, in short NLP methods, and this NLP is well developed research field, especially in terms of deep learning. So for a given sequence, we need to vectorize it in order to apply many kinds of deep learning models, because a sequence is not a numeric values but a series of symbols. There are several ways to vectorize a sequence, such as one-half vector representation and various embedding methods. Once a sequence is vectorized, a sequence input can be fed into any kinds of deep learning models. So effective vector representation method is an essential for the success of deep learning modeling. In the following several slides, we will go over some of the embedding techniques in the NLP domain. I just want to skip the most naive method, one-half vector encoding, because it's straightforward to understand. So I'll start from word2vec, which led the modern innovation in NLP. Then I'll introduce Elmo model, which tries to ameliorate the short length context problem of word2vec. Lastly, we will look into the details of two self-attention based methods. One is transformer and the other one is BERT. Word2vec changed many things in NLP. Long story too short, it opened the deep learning world in NLP domain. It is a word representation where each word in a sentence is transformed into a corresponding vector. When calculating a vector, it considers the local window context. For example, when you want to get a vector for the red word token in the figure, then 4 context words should be considered. And these context words are those blue words in the figure, 2 from the left and the 2 from the right. So, how can you train word2vec? If we have an example sentence like, I go to Emory University, then the output of the model is a target word for example, to. The input is the context words on the both sides, left and right. So we now have the two context words on the left, I and go, and the two on the right, Emory and University. We can create many input and output pairs for any sentences in the dataset. Using those pairs, we can train any deep learning models, and then from the trained network, we can extract the final embeddings, that is a set of vectors for each word in the vocabulary. Although word2vec was so innovative in NLP, it has downsides as the length of the sequence is getting longer. The major reason of this problem is that word2vec only consider fixed length window as context words. Let's think about a situation where the local context is not enough. The top figure shows word2vec. We want to represent the red word, and I put X marks that is closely related to the target word. As you can see, one of the marked word on the far right side that is not considered when representing the target one. This causes information loss. To solve this problem, Elmo is proposed. As you can see in the figure below the word2vec, Elmo considers all words in a sentence, including all related words, to represent the target word. It uses two recurrent neural network models. One of them considers all words on the left, and the other one considers all words on the right. So this solves at least the problem of word2vec. However, we face another problem called information vanishing problem. We have another figure that shows Elmo. There are some differences compared to the Elmo figure in the previous slide. As the distance between the target token and the current token increases, the color becomes lighter. The intensity indicates retained information. For example, the information of the rightmost token or the leftmost token is much diluted when compared to the token immediately adjacent to the target token. This phenomenon is due to the characteristic of the recurrent neural network. As gradually update the information from a far distant token to an adjacent one, the oldest information is getting weaker because it's kept updated. We now have another problem, but people proposed a new model to fix this. That is the transformer. The paper title of the transformer is Attention is All You Need. As you can see from the title, it replaces embedding and recurrent neural network with the transformer. Just like Elmo, transformer also considers all tokens in a sentence. However, it's free from the information vanishing problem because it doesn't use RNN at all. So let's dig into the details of the transformer. The primary purpose of the transformer in terms of an application is to solve machine translation problem. In the figure, the model takes French sentence Je suis étudiant, which is I am a student in English. Then the model translates it and output an English sentence, I am a student. Although the whole model is trained for machine translation, the trained submodel can be used for a word representation task. That submodel is the encoder part of the whole transformer network. The top figure shows the whole transformer network, and it roughly consists of two parts, encoding and decoding. In this talk, we are interested in only the encoding part. So the encoding layers consist of many encoder modules, and each encoder module is composed of a self-attention layer and a feed-forward layer, as shown in the bottom figure. Let's take a closer look at the encoder module. This figure shows how an input is processed by the encoder module, specifically a self-attention layer and a feed-forward layer. Let's say we have two words as an input, A and student. Those are labeled as X1 and X2 respectively. So X1 and X2 are vectors randomly initialized at first, and they are the input of the self-attention layer. The output of self-attention layer is another set of vectors, Z1 and Z2. These vectors are again an input to the feed-forward layer that produces the final output of the encoder module. Since the output of the encoder module has the same format of the input of it, we can stack as many encoders as we want on top of each other. So now, what is self-attention and what is feed-forward layer? The first one is self-attention layer. Let's start with a simple conceptual explanation and then go into more detail. This figure shows the input vector and a self-attention layer and the output vectors. If we want to calculate the new vector for the token student, we just weighted sum, in other words, attention, of all words in the same sentence. That's where the name self-attention comes from. In the figure, the different thickness represent the magnitude of attention. So the word student should get more weights from the closely related words like I and student than other words. So it can be seen as special form of weighted sum. So how can we get those weights and the resulting vectors? Let's say we have a sentence saying drug discovery talk. As I said, the output is also drug discovery talk, but the input vectors and output vectors should be different. The first thing is to calculate value vectors from the token vectors. A value vector is a projection result that will be used in the weighted sum. A value vector is the vector to be summed when the weighted sum is performed and is obtained through a projection from the token vector. In this figure, that projection is done by value matrix, the green matrix, and the resulting value vectors are represented as green vectors. So if we want to calculate the new token vectors, for example, in the figure, a new vector for the token drug, then it's nothing but a weighted sum of the value vectors using the weights W11, W12, and W13. We can do the same thing for the next token discovery. So the resulting new token vector is another weighted sum of the value vectors using another set of weights W21, W22, and W23. The weights are different from the previous example. And the same thing for the last token, talk, using different weights. So now the question is, how can you get those weights? Let's go back to the first token vector calculation example. We know how to get the special vectors, that is value vectors. So we need to know how to get W's. To do this, we need another two more set of special vectors. Those are query vectors and key vectors. A query vector is the projection result using the query matrix, and a key vector is the projection result using the key matrix. Since we have three special vectors, v, q, and k, let me just change the figure so that I can display all of them. Now we have three special vectors for each token. For example, for a drug token, we have Q1, K1, V1, and for discovery token, we have Q2, K2, V2, etc. The query vector is used by the target token, while the key vectors are used for all tokens. For example, if the target token is the first word, drug, then the query vector is Q1, and we are relating Q1 to all of the tokens in a sentence using key vectors K1, K2, and K3. On the left, we can see an example calculation among those vectors. So W11 is the inner product of Q1 and K1. W12 is the inner product of Q1 and K2. And W13 is the inner product of Q1 and K3. So if the dot product results are just like the numbers shown, we can use those numbers as weights when we do the weighted sum for the target word drug. So the new token vector of the target, Z1, is the weighted sum of value vectors W11V1 plus W12V2 plus W13V3. We can repeat the similar process for the other two tokens, then we can get Z2 and Z3. So this is the summary of one self-attention. For given input vectors, we get three special vectors: value vector, query vector, and key vectors. By using the dot product, we can get the output vectors. This whole process is done in one head. If we do this process several times, like 8 times, for the same input, then we can have 8 different sets of output vectors. Those repetition is called as multi-head operation. If there are 8 heads, then we need to have 8 different sets of parameters for the special vectors. The reason why we need more than one head is that we are expecting each head to learn different aspects. As a result, we can have richer representations. Another layer of one encoder module is a feed-forward layer. As described so far, we have 8 sets of Z's from the 8-headed self-attention layer. We concatenate them together so that it can create a red matrix in the figure, and this is the input to the feed-forward layer. We have another trainable parameter presented as green matrix, and we multiply the input with this matrix, so we get the output vectors shown as blue matrix. These vectors are the final output of one encoder module, and it becomes the input to the next encoder module. Up to this slide, we learned about the encoder module in the transformer model. The next topic is about the potential problem of self-attention layer. to do that, so we go back to the figure that explains the weighted sum of the first word. Since this is the weighted sum, the resulting vector doesn't take account the order of words. For example, the self-attention output vector of drug discovery talk will be the same as the self-attention output vector of talk discovery drug. And this is the case where we get a low quality representation because the latter sentence makes no sense, but the output of it is the same as the former one. To handle this problem, the authors of the transformer add positional encodings to the input vectors. It's shown in the figure. Yellowish vectors are the positional encodings, and they are added to the inputs. The positional encoding is a fixed pattern depending on the position of each token in a sentence. Therefore, a model can recognize each token's relative location in one sentence. Okay, up to this point, we looked at the transformer model, and we are ready to proceed to BERT. BERT is short for bidirectional encoder representations from transformers. This is proposed by Google AI team in about 2 years ago. As shown from the citation count of almost 10,000, this research has been so impactful in many deep learning communities. In the original paper, it was the state of the art on 11 NLP tasks, and for the question answering task, which is called squad, it even outperforms the human level. In terms of the model, it adapts the encoder module of the transformer and adds 2 new language model tasks. So here is the overview of the model. In the BERT, the input can be at most 2 sentences. So they add another encoding to the input. And for the task, in the pre-training step, they use 2 tasks, the mask language model task and the is next task. I will explain these things one at a time. Firstly, new input representation. This figure is adopted from the BERT paper. It presents 2 major modifications of input compared to the transformer. The first modification is by adding special tokens. CLS token, that is in the first location of the input, it's used for classification task after pre-training. Another special token is SEP, and it's used to mark the end of a sentence. Another modification is that they add segment embedding, where we add zeros to the first sentence and add ones to the second sentence. This could be more informative for a model to distinguish the first sentence from the second one. So next is one of the 2 pre-training tasks, that is masked language model, in short MLM. It's known that the major performance improvement is attributed to this new task. Briefly speaking, MLM is a task where some of the input tokens are masked and the model predicts what were the original tokens for those masked tokens. For example, if the original sentence is I am a student, then we randomly select a token. Let's say we choose M, and replace it with mask token and save the location of the mask token, which is position 1. Then this masked sentence and the location information will be the input to the pre-training model. If the model predicts the original token M, it'll get 0 error signal. Otherwise, it will get an error signal so that the network can be updated properly. From this task, the model will learn syntax and semantics together, but this MLM task, as it is, has a downside. Mask token is used only in the pre-training, so it's never seen during fine-tuning. That is directly related to the target task. So the authors mix up random tokens with mask token. Another problem is the training time. Because of enormous training time requirement, they just masked 15% of the whole token in the training set. With this reduced setting, they spent 4 days with the most powerful TPU machine at that time. If we do this using Google Cloud credit, it will require more than $10,000, which is very expensive for just one training. Another new task of BERT is next task. This task was suggested to enable the model to learn the context of 2 consecutive sentences. As we saw, the input consists of 2 sentences, so if these 2 sentences are in a consecutive order, then the model should produce 1, otherwise it should output 0. So it's a binary task, and the dataset can be automatically generated just like MLM task. Therefore, these 2 tasks can be seen as self-supervised learning. With these model details, they experimented 2 models, the light model called base and the heavy one called large. It differs in terms of the vector dimension, the number of heads, and the number of layers. The large model is about 3 times bigger than the base model. After pre-training these models, they fine-tune the model differently according to the target task. The authors fine-tuned the model for 11 tasks, but those tasks can be divided into 4 groups depending on the task types. The first task type is sentence pair classification. Since this is a classification task, we use the special token we learned, CLS token. So we put a dense network on top of this CLS token. So for example, if the task is to predict if given 2 sentences are semantically equivalent, then the output of the dense network attached on the CLS token will be 1 if the input are equivalent, or 0 otherwise. Using pre-trained weights, we fine-tune the network with this dataset. The next task group is the single sentence classification. Again, this uses the CLS token because it's a classification. And this group takes only 1 sentence as an input. An example task is a sentiment classification task called SST. The input is a movie review and the output is sentiment label of that input. The third task group is the question answering. Unlike the previous two task group, this group takes two paragraphs as an input, not sentence. So one is a question and another one is a paragraph that includes the answer for that questions. The special token SCP can be used to mark not only the end of the sentence, but also the end of a paragraph. For example, if the question is, "Where is Emory University?" and the paragraph is, "Emory University is a private research university in the Druid Hills neighborhood of the city of Atlanta, Georgia, United States," then the correct answer is Druid Hills neighborhood of the city of Atlanta, Georgia, United States. Then the output is the first token and the last token of the answer, which is Druid and state. So we put a classifier on all of the tokens output so that it can predict either start of the answer or the end of the answer or nothing. The last task group is single sentence tagging. And the only task in this group is named entity recognition, in short NER. NER is to find any named entities in a sentence. A named entity could be either organization, people, or location. So the example of organization is Deargen or some NVIDIA or some organization, and people could be some named person, for example, Dr. Shin or Bill Gates or something. Well, location could be a known location like Seoul or Atlanta or something like that. So, for example of any other task, if the given sentence is Dr. Shen is a co-founder of Deargen, then we tag non-entity token with the symbol O and people entity token with the symbol PER and organization entity with the symbol ORG. So this is token-wise classification, so we put a classifier on all of the tokens. So up to this slide, we learned various sequence representation method, especially in NLP domain. Now, we finally came back to the drug discovery problem. Before Deargen's model, DeepDTA was the best model in the drug-target interaction problem. They just used one half-vector embeddings and applied convolution and dense network to get a prediction of the affinity score of the given two input. So let's take a look at each operation. If we apply convolution operation on a molecule sequence, we convolute several tokens in a sequence as shown in the figure. So for example, in the figure, 12 tokens are marked as red, and those 12 tokens are convoluted together and produces one number, let's say 5. Then we slide one token right. We apply another convolution on that shifted 12 tokens, and we get another number, say 3. Another convolution with one more shift, we get 8. And we repeat this to the end of the token. Then we can get a feature vector for a molecule. We do the same thing for the protein sequence. and we get a protein feature vector. We then concatenate these two feature vectors and pass it through multiple dense layers, and it will produce the final affinity score. This is how deep DTA works, and this is the first deep learning method in the DTI task, which shows the promising result. However, it has a limitation because of the characteristics of the convolutional operation. A molecule actually is three-dimensional, as shown in the graph, but the model uses a one-dimensional sequence. So if they apply convolution, then it only considers local context, while actually global context might be important. For example, F and Cl atom in the sequence, they are represented with green in the graph and the sequence. They are very near together in a sequence, so they will be convoluted together, but they are actually in long distance, as can be verified, green atoms in the graph. Let's look at another pair, Cl and N. They will never be convoluted because they are far in distance in the sequence format. However, its actual distance is closer than F and Cl pair, so they might contain important information. This example shows the importance of global context, and this is the motivation of Deargen's model. So this is molecular transformer that is proposed by Deargen. It adapts BERT architecture with some modifications. First modification is that we use characters as a token while BERT uses word. Secondly, we modified special tokens to effectively model a molecule sequence. Lastly, we use only MLM task because we don't use context of multiple sequences. This is how we process the input molecule sequence. With some positional encodings and molecule token embeddings to get the final input of the model. And we add two special tokens, begin and end, so that it can mark truncated sequences. In order to reduce the model complexity, we fixed the sequence length as 100. So as shown in the example below, if a sequence is longer than 100, then we truncate it and put those special tokens, the begin token at the start of the sequence and end token at the end of the token. We pre-train this molecular transformer using 19 millions of molecules in the PubChem database. The network has 8 layers, 8 heads, and 128 vector size. We trained this model using 8-core TPU for about 60 hours. As a result, we got about 97% of accuracy in MLM task. Compared to the NLP case with BERT, which recorded 98%, we thought it is a reasonable pre-training, so we transfer these trained weights to the main DTI model. The main DTI model, which is called MTDTI, it consists of 3 parts. The molecule representation part in red, the protein representation part in blue, and the interaction modeling part in green. and we name it as MTDTI. For the protein representation, we use CNN because of the low accuracy in MLM task. Um, Only the parameters of the red part will be initialized with the pre-trained weights, and other two parts will be randomly initialized. To effectively evaluate the result, we used four metrics. C index is the probability of being correctly ordered of two random samples. MSE is the general loss term. Matrix used in QSAR study is measuring the correlation between the goal values and the predicted values. And lastly, for AUPR, we changed the problem as binary classification and measured the general performance of it. After fine-tuning the model using two public DTI datasets, Kiba and Davis, we get this result, a new state-of-the-art in DTI. It outperforms all other methods in all four metrics. The interesting point is that if we don't fine-tune the empty part, it still gives competitive results. Those results are presented as empty DTI without fine-tuning. This result indicates that MT actually produces very useful representation that can be directly used in the DTI task. To further verify the promising performance of the MT-DTI, we designed two case studies. The first case study is to find potential drug candidates for a specific cancer-related target, EGFR protein. So we fixed the target protein as EGFR, and we ran the trained model with 1800 FDA-approved drugs. Then we get 1800 affinity scores as the model output. We then sort them in descending order. The result was very interesting. That all 8 known EGFR drugs are listed in our top 30 list shown in the table. All those known drugs are marked as bold and with a star. This promising result could indicate other non-EGFR drugs might possibly be a new anti-cancer drug candidate. The second case study is about COVID-19. Similar to the previous case study, we fixed the target as the proteins of the novel coronavirus, and we ran the trained MTDTI model with 200 antivirus drugs. And then we put a short list of candidate drugs, including remdesivir, even before the onset of the clinical test of remdesivir. Therefore, our model successfully predicted the efficacy of remdesivir using only in silico method. So far in this talk, we learned the basics of drug discovery, and we reviewed some of the NLP methods that are closely related to the drug discovery model, and showed how a deep learning model can be used to discover a new drug candidate. Lastly, by showing two case studies, we confirmed that this approach is promising and it's worthy to research more. I hope this talk helped you to understand deep learning and drug discovery. Thank you very much.